Okay, my dear student, I am Dilip Kumar welcoming you to this beautiful series. And today we are going to discuss about the stamen or microsporophyll. Stamen. Stamen made up of two parts. Here I am just going to draw the structure. Here is an anther and here is the filament. And these united with each other to form stamen. So in sense, stamen has two parts. Okay, one is anther and another is filament. Okay, anther here you can see the anther is bilobed. Okay, and these two lobes are connected with each other by a sterile connective tissue. So anther is bilobed and these two lobes are connected with each other by a sterile connective tissue. Sterile connective tissue. Why it is called sterile connective tissue? Because it does not take a part in the process of reproduction. Its only function is to hold the two lobes close to each other. If we dissect the anther at its transverse section, you can see I am just going to dissect the anther at its transverse sections. Okay, you can see. Okay, you can see. Okay, that is the anther. I have dissected it at its transverse sections. So you can see these two lobes and it is the sterile connective tissue. Okay, it is the sterile connective tissue. It is the sterile connective tissue which connect these two lobes with each other. Okay, you can see there are four groups in anther. 1, 2, 3, 4. Okay, these groups are called theca. These groups are called theca. So in a anther there are four theca, 4 in number. In anther there are four theca, and in one lobe there are two theca. One lobe contain two theca. So it is called bilobed. Dithecous. So one lobe contain two theca. So it is called dithecous. It is called dithecous. Here we get in conclusion that it is bilobed, the dithecous. Okay, and in two lobe contain four theca, and you can see in each theca contain a microsporangia. Okay, and each theca contain a microsporangia. So in a anther there are four microsporangia. So here we get in conclusion that one anther, two lobe, okay, means bilobed. Okay, one lobe, two theca, one theca, one microsporangia. So four theca, four microsporangia, and so in one anther, four microsporangia. Okay, get it? So these four microsporangia gives pollen sac at a mature stage of anther. So we can compare here in between the mature and unmature anther. Okay, so unmature anther and mature anther. Okay, in unmature anther, this is called, here you can say this is called microsporangia. Okay, in unmature anther this is called microsporangia. Okay, which is diploid in a number. Okay, in mature anther, this microsporangia gives rise to microspore mother cell, MMC, microspore mother cell, which is a diploid number, and in mature anther it gives rise to pollen grain, okay, which is haploid in number, and it is by the process of meiosis, meiosis. It is in the process of the division here occurs is meiosis. Okay, my dear students. Okay, so you can see one mature anther is equal to tetrasporogenate. Okay, why it is called tetrasporogenate? Here four microsporangia gives rise to 4 pollen grains, so it is called tetrasporogenate. Okay, now I am going to show you the more dissect and analytical section of anther, that is a TS of anther, transverse section of anther. Okay, you can see, if I show you under the microscope, you can see here is the okay, here is the sterile connective tissue, and here is the microsporangia. You can see here is the microsporangia, and you can see there are more layers present around the particular structure. Here you can see another more layer present around the structure. Okay, you can see, and also there is a structure present around the microsporangia. So so in total you can calculate there are 4 layers, 1 2 3 4 layers. Okay, this first layer, this is the one layer, the first layer, considering from the outside, the first layer is called exothecium or epidermis. Okay, and the second layer is called endothecium, and the third layer is called middle layer. Okay, and the fourth one is called tapetum. Okay, tapetum. I am just going to, that is the exothecium, second one is the endothecium, third one is the middle layer, and fourth one is here. Okay, and the fourth layer, which is called tapetum, is attached to the microsporangia. Okay, so there are 4 layers present in anther around the microsporangia. Okay, now we are just going to discuss all these layers. I am just going to draw the another diagram of the transverse section of anther, that is equal to, here you can see, I am just going to draw in large. Okay, you can see here is the sterile connective tissue. Okay, you can see here is the microsporangia present. You can see there is a single layer of flat cells. There is a single layer of flat cells present at the outer side or first, that is called exothecium. Here is the exothecium, that is the first layer. The first layer is equal to exothecium or epidermis. Okay, so what is it? You can see these are of looks like flat cells. Okay, single layer, these are a single layer. Okay, and looks like structural like flat cells. What type of a cell it is? It is a parenchymatous cell. It is a parenchymatous cells. Okay, function of that cell is the protection is the function. Okay, and the second layer, then the second layer is you can see there is a radial layer of cells, a layer of radial cells present. You can see there is a layer of radial cells present. Okay, you can see these radial layer of cells are called second layer, and this second layer is called what? These second layer are called endothecium. Okay, endothecium, it is made of radial cells. Okay. Single layer, it is also present in single layer. Okay, and it is made up of or it is supplied with the alpha cellulosic fibrous band. Okay, so what is the functions? It is hygroscopic in nature. Hygroscopic in nature means it absorb water. When the anther gets mature means when after the formation of pollen grains, the dehiscence occurs. It is the dehiscence site. Here is the dehiscence site. Here is the dehiscence means the rupture of anther. Dehiscence means the rupture of anther. The rupture of anther occurs and the pollen grain comes out from the anther in order to bring out the pollinations. Okay, alpha cellulosic fibrous is hygroscopic in nature and it absorb water, and as a result the anther becomes swell off and dehiscence occur at this site. Here is the site, dehiscence site. I can show you by take the right. Okay, so it helps in dehiscence. When dehiscence after mature of anther. Okay, so before maturation, what is its function? Before maturation, its function is protection before mature. Okay, third one is middle layer. You can find there are 2 to 3 layer of cells are present. Okay, here is the one layer and here is the second layer. You can find. Okay, and you can find is the third layer. Okay, you can find there are 2 to 3 layer of cells are present. These are called middle layer. Okay, so middle layer, it is the third is the middle layer is nothing. It is a 2 to 3 layer of cells. 2 to 3 layer of cells. Okay, so it disappear quickly after getting mature of anther. It disappear quickly means from these 3 layers, 1 2 3 layers, it disappears fast. Okay, middle layer has a shortest lifespan, means it disappear fast. Okay, it disappear. Function of a middle layer is protection. Okay, last one is tapetum. Okay, last one or fourth layer is tapetum. Tapetum is a pyramidal cells. Okay, which is completely surrounded it is the layer of pyramidal cells which completely surrounded the microsporangia. Okay, which completely surrounded the microsporangia. Okay, it is a layer of pyramidal cells. Okay, my dear students. Okay, so that is a tapetum. Okay, pyramidal cells. Okay, it is a pyramidal cells. Okay, attached to microsporangia. Attached to microsporangia. Okay, and it has a large cytoplasm. Okay, it's a cytoplasm is highly supplied with nutrients. Why it is supplied with nutrient? Because it provides nutrition to the microsporangia, developing spore or microsporangia. Okay, it provide nutrition. To developing microspore. Okay, or microsporangia. Next, and it has also UBC bodies. Okay, it has also UBC bodies. which secrete sporopollenin. Sporopollenin makes the outer covering of the pollen grain. Okay, outer covering makes outer covering of a pollen grain. Makes outer covering of a pollen grain. Okay, which is called a exine. Which is called a exine. Okay, my dear students, next it also secrete an enzyme. It also secrete an enzyme called callage. Okay, which dissolve the which dissolve a cementing substance present in the microspore, that is callose. Okay, it also helps in formation of a pollen kit. Okay, it also helps in the formation of a pollen kit. Okay, that's all. Sorry, it also helps in the formation of a pollen kit. Like enzyme called callage enzyme, it also secretes the callage enzyme, and it also helps in the formation of a pollen kit.